In the summer of 2025, inside a secure genetics laboratory in London, a team of researchers stared at a computer screen holding data that should not have existed. For over 100 years, every single attempt to pull a complete genetic blueprint out of an ancient Egyptian body had failed, one after another, without exception. Not from a pharaoh, not from a queen, not from any of the thousands of mummies stacked in museum basements around the world. Egypt, the single most obsessively studied civilization in human history, had somehow become a black hole on the genetic map of the ancient world. And then, without warning, a skeleton that had been sitting forgotten on a shelf in Liverpool for more than a century finally broke that silence. And what it revealed sent a wave of shock through the entire field of archaeology. The story begins not in a pyramid, but in a rock-cut tomb chamber carved into a cliffside roughly 265 kilometers south of Cairo in a village called Nuweirat. In 1902, archaeologists working the site pulled a human skeleton out of the ground, still sealed inside a large ceramic burial pot. There was no golden mask, no inscribed sarcophagus, no elaborate mummification at all. Just bones folded into plain fired clay, tucked into the rock high above the Nile. It looked, to the archaeologists digging it up, like an unremarkable burial. Nobody could have guessed that this exact body, out of every mummified pharaoh and noble ever pulled from Egyptian sand, was the one that would finally answer a question scholars had been arguing about for generations. The skeleton was shipped across the Mediterranean and the Atlantic to Liverpool, where it became part of a growing university collection of Egyptian antiquities. Then, in the Second World War, disaster struck. A German bombing raid scored a direct hit on the museum, destroying the overwhelming majority of its human remains collection in a single night. Bodies that had survived nearly 4,000 years in the Egyptian desert were wiped out in seconds by 20th century warfare. This particular skeleton, tucked safely away in storage at the time, happened to survive. And then it simply sat there, largely unexamined, for the better part of 100 years, while the science of genetics raced ahead everywhere else on the planet without ever quite catching up to Egypt. That changed in July of 2025 when a research team led by Adeline Moores-Jacobs of Liverpool John Moores University, working with Pontus Skoglund's Ancient Genomics Laboratory at the Francis Crick Institute in London, published their findings in the journal Nature under the title Whole-genome ancestry of an Old Kingdom Egyptian. The team had done something that had defeated every previous attempt in the entire history of Egyptology. They extracted a complete, usable whole genome from a body that had been dead for close to 5,000 years, a body older than the Great Pyramid of Giza itself. This wasn't the first time someone had tried. Back in 1985, the molecular biologist Svante Pääbo, who would later go on to win a Nobel Prize for his work sequencing Neanderthal DNA, published a paper in Nature claiming to have recovered genetic material from a 2,400-year-old Egyptian mummy. At the time, it looked like a breakthrough. It wasn't. Later, reanalysis showed the sample had almost certainly been contaminated with modern human DNA introduced during handling, and the claim quietly fell apart under closer scrutiny. For decades afterward, that early failure cast a long shadow over the entire idea of recovering ancient Egyptian DNA, and many researchers simply assumed it could never be done reliably at all. Then, in 2017, a team led by Verena Schoenemann managed to extract partial genome-wide data from a handful of mummies at Abusir el Melek, proving it wasn't completely impossible, but even that success came with an asterisk. Every sample they recovered dated from more than 1,000 years after Egypt's pyramid-building age had already ended. The oldest and most formative period of pharaonic civilization, the era when the state itself was being built, remained completely invisible to genetics. Nobody had a genome from it. Nobody knew if one could even survive. To understand why this took over a century and why it frustrated geneticists for so long, you have to understand exactly what makes Egypt one of the single hardest places on Earth to recover ancient DNA from. DNA is a genuinely fragile molecule. Its rate of decay after death depends heavily on temperature, humidity, and burial conditions. Cool, dry, stable environments, places like Arctic permafrost or high-altitude caves, can preserve usable ancient DNA for tens of thousands of years, which is exactly why so much of the field's most detailed genetic work has come out of Siberia and Scandinavia. Egypt represents close to the opposite scenario. The same relentless heat that helped desiccate and preserve Egyptian bodies, art, and papyrus for millennia was simultaneously destroying the one thing scientists needed most, actively shredding DNA into fragments too short and too damaged to ever be reliably sequenced. And it gets worse. The very process that Egypt became famous for, artificial mummification, using resins, natron salt, and other chemical treatments to halt bodily decomposition, introduced additional contamination that degraded genetic material even further. Harold Ringbauer, a population geneticist at the Max Planck Institute for Evolutionary Anthropology, later explained that samples embalmed using standard mummification techniques turned out to be far poorer sources of usable ancient DNA than researchers had hoped. precisely because the embalming process worked against genetic preservation even as it kept the visible body looking remarkably intact. In other words, the more spectacular and famous the mummy, the less likely its DNA had survived. For over a century, that left the deepest, most fundamental question about the people who built the pyramids, where their ancestors actually came from at a genetic level, essentially unanswerable no matter how many tombs archaeologists cracked open. That gap put Egypt in a strange position within the broader science of ancient genetics. Researchers had already built increasingly detailed genetic maps of ancient Europe, the Eurasian steppe, the Americas, and other parts of Africa, tracing migrations and population mixing back tens of thousands of years using recovered ancient DNA. In some cases, those genomes were even older than anything Egypt had produced. Yet Egypt, arguably the single most intensively studied ancient culture on the planet, with more written records, art, and archaeology than almost anywhere else, remained comparatively dark on that same genetic map. Its history was rich beyond measure. Its actual population genetics, at the deepest level, were essentially unknown to science. That gap left one particular historical question stuck in limbo for decades. Egyptologists already knew, from pottery, artifacts, and administrative records, that Egypt had significant contact with the wider Fertile Crescent region to its east, the area covering Mesopotamia and the Levant, well before and during the rise of the pharaonic state. Domesticated plants and animals, certain administrative practices, symbolic parallels, and crucially the potter's wheel itself, all show clear signs of spreading from Mesopotamia into Egypt during the 4th and early 3rd millennia BCE, in a period archaeologists call the Uruk expansion, named after the great Mesopotamian city whose cultural influence seems to have radiated outward across an enormous stretch of the ancient world. But here's the problem that had frustrated scholars for generations. Shared objects and technologies cannot by themselves tell you whether real people migrated and mixed together, or whether separate populations simply borrowed clever ideas from their neighbors through trade. Pottery styles and symbolic systems cannot prove that human beings, rather than just their goods and their ideas, actually made the journey between these two early centers of civilization. Only a genuine ancient genome, recovered from an individual old enough to have lived near the actual period of contact, could settle the question directly. And until 2025, nobody had ever managed to recover one old enough to test it. Even the location of the tomb itself carried a kind of dark luck. Nuweirat sits inside a limestone cliff face, and rock-cut chambers like this one tend to hold a far more stable temperature than the open desert sand where so many other burials were placed. Combine that stable microclimate with the sealed ceramic pot surrounding the body, and you get a rare double layer of protection, one that most Egyptian burials simply never had. It's the kind of accident of geography and circumstance that no ancient Egyptian could have deliberately engineered for the benefit of scientists 5,000 years in the future. And yet, it is exactly what made this discovery possible at all. Now for the part that shocked the researchers themselves. Using cementum tissue, a dense mineralized layer at the roots of two teeth that shields DNA from environmental damage far better than bone does, the team sequenced the man's genome to roughly twofold coverage inside strict cleanroom protocols designed to keep modern contamination out entirely. They then compared his genetic profile against a reference library of thousands of ancient and modern genomes spanning Africa, the Middle East, and Europe, statistically modeling exactly which ancient populations best explained what they had found. The individual, now known in the scientific literature by the code name NUI-ZSRO1, was radiocarbon dated to between 2855 and 2570 BCE, placing his life right at the boundary between Egypt's early dynastic period and the Old Kingdom, the exact era when Egypt's earliest pyramids were beginning to rise and the pharaonic state was still consolidating its power. Before this discovery, the oldest and most complete genetic data available from ancient Egypt came from a landmark 2017 study analyzing 90 mummies from the site of Abu Sir el-Melek, remains dating from more than 1,000 years later, spanning the New Kingdom through the Roman era. Even that earlier study had only recovered partial genome-wide data, and from just 3 individuals. NUI-ZSRO1 didn't just add one more data point. He pushed the genetic record of ancient Egypt back over a millennium into a period for which no comparable genetic evidence had ever existed. And what his DNA revealed answered the Mesopotamia question with a level of directness that stunned even researchers who had long suspected a connection existed. Rather than showing a purely local North African genetic profile, which some earlier models of Egyptian population history had assumed given the region's deep archaeological continuity, the analysis found that his ancestry was best explained by 2 distinct sources. Roughly 78% of his genetic ancestry traced most closely to Neolithic populations of northwest Africa, specifically to genomes recovered from the middle Neolithic site of Skirat Rawazi in modern Morocco, dated to somewhere between 4780 and 4230 BCE, representing the deep indigenous North African population that had inhabited the region for thousands of years before written Egyptian history even began. But the remaining slice of his ancestry, roughly 20 to 22%, depending on the statistical model used, traced clearly and specifically back to populations from the Eastern Fertile Crescent, showing the closest genetic affinity to Neolithic Mesopotamian farming communities, dating all the way back to between 9000 and 8000 BCE. Communities from sites including Nemrik and Bonkuklu Tarla in what is now northern Iraq and southeastern Turkey, among the very first places on Earth where humans domesticated crops and settled into permanent agricultural life, laying the foundation for civilization itself thousands of years before the first Egyptian pyramid was ever conceived. This finding did more than confirm some vague genetic link between ancient Egypt and Mesopotamia. It provided for the first time direct biological proof that actual people, not merely trade goods and technological ideas, had physically moved between these two centers of early civilization and interbred with the population already living in the Nile Valley generations before this man was even born. Pontus Skoglund of the Francis Crick Institute put it plainly after the study's publication, describing the discovery as the first genetic evidence that people had moved into Egypt from the Fertile Crescent and mixed with local populations at this early point in Egyptian history. It confirmed, through hard genetic data, exactly what archaeologists had only ever been able to infer indirectly from shared pottery and administrative practices without ever being able to prove it. Here is where the story takes an even stranger turn. Isotope analysis conducted on NUE 0-1's own teeth, which can reveal information about the climate and geography a person grew up in based on chemical signatures absorbed from local food and water during childhood, confirmed that he himself was not a first-generation migrant from Mesopotamia. His isotopic signature was fully consistent with a childhood spent in the hot, dry environment of the Nile Valley, eating a typical local diet of wheat, barley, terrestrial animal protein, and likely Nile fish, common staples for Egyptians of the period. In other words, the Mesopotamian genetic contribution inside his genome hadn't arrived with him personally. It had already been present in the Egyptian population for some number of generations before his birth, the product of an earlier wave of migration and mixing that predated him entirely. That single detail forced researchers to push the true timeline of this migration back even further, to somewhere around 2700 BCE or earlier, a period the genetic record had never previously been able to reach at all. It quite possibly connects directly to that same broader Uruk period cultural exchange archaeologists had already identified through pottery and administrative technology, now finally confirmed as involving real human migration, rather than cultural borrowing alone. The physical evidence recovered alongside his DNA added a genuinely human dimension to an otherwise abstract genetic percentage. Bioarchaeologist Joel Irish's detailed skeletal examination found that NUE-001 stood approximately 160 centimeters tall and died somewhere between the ages of 44 and 64, a genuinely advanced age for a period when life expectancy ran considerably shorter than it does today. His skeleton showed pronounced osteoarthritis in his knees, hips, and right foot, along with expanded seat bones and extensive repetitive wear patterns in his arms, a combination of physical markers. Irish concluded were consistent with decades spent working at a potter's wheel, seated for hours at a time performing the same repetitive hand and arm movements throughout his working life. There is a quiet irony worth sitting with here. The same potter's wheel technology that had itself spread into Egypt from Mesopotamia during the same broader period of cultural contact may well have been the very craft this particular man, carrying Mesopotamian ancestry inside his own genome, spent his entire working life practicing. His own body's inheritance and his daily trade were connected in a way neither he nor anyone around him could ever have known. The circumstances of his burial add one final layer to the picture, and this is the detail that ultimately saved his DNA from destruction. Despite the physical evidence of a working life spent in demanding manual labor, NUE-01 was buried with what researchers interpreted as clear signs of elevated social status. His body was placed intact, without the resin and chemical treatments of formal mummification, inside a large ceramic vessel, which was then sealed within a rock-cut tomb chamber rather than a simple pit grave. That level of mortuary care suggests genuine respect and honor, even for a man whose bones tell the story of decades of hard physical work. Researchers flagged this combination, evidence of manual labor alongside an elevated burial, as a plausible sign of social mobility within Old Kingdom Egyptian society, a hint that exceptional skill in a trade like pottery could elevate a man's standing well beyond what his manual occupation alone might suggest. And this exact style of burial turned out to be the direct reason his DNA survived at all. Because he died before mummification became the dominant Egyptian burial practice, His body was never treated with the resins and preservatives that destroy genetic material in later mummies. Meanwhile, the sealed ceramic vessel and the cool, stable microclimate of the rock-cut chamber protected his remains from the temperature extremes that shred DNA in more exposed burials. The absence of the very embalming process Egypt would later become world famous for is, somewhat ironically, the specific reason this became the oldest successfully sequenced genome from ancient Egypt, rather than one of the countless mummified pharaohs and nobles whose far more elaborate, far more famous remains have so far yielded no comparable genetic data at all. It's a reminder that the most historically celebrated burials are not always the most scientifically valuable ones. Researchers also compared NUE-Zoroastrian's genome against the genetic data recovered from the earlier 2017 Abusir el-Malek study, whose 90 mummy sample dated to well over 1,000 years after his death, spanning the New Kingdom through the Roman period. That comparison found only marginal genetic continuity between the two populations, alongside clear evidence of a further, more substantial infusion of Levantine ancestry entering the Egyptian population sometime between NUE-Zoroastrian's era and that later period, with Bronze Age Levantine ancestry accounting for roughly 64% of that later population's genetic makeup in the best-fitting statistical model. The same 2017 study had also found that modern Egyptians today actually carry somewhat more sub-Saharan African ancestry than either the ancient Abusir el-Malek mummies or NUE-Zoroastrian himself, a shift researchers attribute to increased mobility along the Nile, expanded long-distance trade with sub-Saharan Africa, and the trans-Saharan slave trade that intensified over roughly the past 300 years. It's worth pausing on just how significant that 64% figure really is. It means that in the roughly 1,500 years separating NUE-Zoroastrian from the mummies of the New Kingdom and Roman era, the genetic makeup of the people living along the Nile had shifted dramatically, and not through some slow trickle of contact, but through what looks like a substantial and sustained wave of movement from the Levant. That kind of shift lines up with everything historians already know about the sheer intensity of Egypt's later foreign entanglements, from the Hyksos period, when rulers of Levantine origin came to control parts of northern Egypt, through the New Kingdom's extensive military campaigns and trade networks, stretching deep into modern-day Israel, Lebanon, and Syria. Genetics, for the first time, put a number on a process that historians had only ever been able to describe in general terms. Together, these two studies, separated by more than a millennium of Egyptian history, sketch a genuinely dynamic picture. Ancient Egypt was never one single founding population that stayed genetically frozen from the earliest pharaohs through the Roman conquest. Instead, it experienced multiple distinct waves of contact and mixing with populations from the wider Fertile Crescent and beyond, stretching from the Neolithic period detected in NUE-Zoroastrian's own genome through later Bronze Age movements reflected in the more recent mummy samples, and continuing still further with the sub-Saharan contributions visible in Egyptians living today. Egypt, in other words, was never the sealed, unchanging genetic isolate that some earlier and simpler models of the ancient world had assumed. From its earliest recoverable genetic moment, it was already a crossroads. For more than a century, and as the skeleton of a man who once spent his working life bent over a potter's wheel sat quietly in a museum in Liverpool, having survived a transatlantic shipment, a world war, and the near-total destruction of the collection housed around it, without anyone realizing what his bones were actually capable of revealing. It took modern genome sequencing technology, a fortunate accident of burial circumstance that spared his body the chemical preservation that would have destroyed his DNA, and a research team specifically hunting for the oldest viable genetic sample Egypt had to offer, to finally let one ordinary craftsman answer a question that a century of archaeology, pottery analysis, and administrative record-keeping had only ever been able to guess at. The scale of what this single genome accomplished becomes even clearer when you compare it to the broader global picture of ancient DNA research happening at roughly the same time. In the 2 decades before this study, geneticists had already pulled complete genomes out of Siberian permafrost specimens tens of thousands of years old, out of Bronze Age skeletons scattered across the Eurasian steppe, and out of Ice Age hunter-gatherers buried in European caves for over 30,000 years. Compared to all of that, a genome dated to roughly 2700 BCE doesn't sound particularly ancient at all. But for Egypt specifically, it represented something close to a miracle, simply because the country's climate and burial customs had made recovering anything at all from that deep in its history seem almost impossible. The contrast says less about how old NUE-Zoroastrian's genome actually is, and more about just how uniquely hostile Egypt's environment has always been to genetic preservation, compared to almost anywhere else ancient DNA has ever been successfully recovered. Not whether cultural ideas moved between Mesopotamia and Egypt at the dawn of pharaonic civilization, but whether actual human beings did, carrying their ancestry with them into the deep genetic history of one of the world's great civilizations. There is something worth sitting with in the fact that this answer came not from a pharaoh, not from a queen, not from any of the famous mummified rulers whose faces and names we already know from millennia of Egyptian history and countless documentaries, but from an anonymous working man buried without embalming, whose aching joints tell us more about how he actually spent his days than any inscription ever could. The grandest and best preserved monuments of ancient Egypt, ironically, turned out to be genetically silent, their own mummification chemistry destroying exactly the evidence modern science needed most. Meanwhile, an unassuming potter's burial pot, sealed by circumstance rather than design, held the oldest voice ancient Egypt has left us. A voice that, when finally read at the level of the genome, confirmed something written in his bones and blood long before any hieroglyph was ever carved to record it. Two civilizations that history books often treat as separate, self-contained stories, Egypt rising along the Nile, Mesopotamia rising between the Tigris and Euphrates, turn out, at the genetic level, to have already been quietly intertwined by the time Egypt's first pyramids broke the horizon. And there is a bigger, slightly unsettling question hiding behind all of this. If a single grave excavated in 1902 and left largely unexamined for over a century could still hold an answer this significant once the right technology finally caught up to it, how many other quiet, ordinary burials, sitting in museum storage rooms around the world right now, completely unremarkable to look at, are waiting for their own moment to rewrite what we think we already know about the deepest chapters of human history? Scientists have only just proven that Egypt's DNA can survive at all, under exactly the right conditions, and that means the oldest chapters of the human story may still be sitting quietly on a shelf somewhere, forgotten, waiting for someone to finally look. What makes this whole story even more remarkable is how narrow the window of opportunity actually was. Nuzuzuzuru I died at exactly the right moment before mummification became the standard practice that would have destroyed his DNA, and he was buried in exactly the right kind of tomb, one that happened to combine a stable rock cut chamber with a sealed ceramic vessel. Move his death forward a few centuries, into the era when embalming had become universal, and his genome would almost certainly have been lost forever, just like the genomes of the pharaohs who came after him. Move his burial to open desert sand instead of a shaded cliff chamber, and the heat alone might have finished off what little DNA remained. It took an extraordinarily specific combination of timing, geography, and burial custom, all lining up by pure chance for his bones to still be carrying a readable genetic code 5,000 years later. That narrow window is exactly why researchers are now racing to find other candidates like him, other unmummified burials from Egypt's earliest dynastic period, sealed away in similarly protective conditions, that might still be sitting untouched in museum storage collections around the world. Liverpool's own collection alone contained thousands of items pulled from Egyptian soil over more than a century of excavation, most of it never subjected to modern genetic analysis at all. If one accidental survivor from 1902 could rewrite the earliest chapter of Egypt's genetic history, the obvious next question is, how many more are still out there, waiting quietly in drawers and storage crates, unlabeled and unremarkable, for a research team with the right technology and the right question to finally come looking, and for another silent voice from the deep past to finally get the chance to speak?